Hello students, welcome to LEELA'S tutorial. Let us discuss about generation of gene knockout mouse. What do you mean by gene knockout mouse? So here it is an laboratory animal where the researchers have either inactivated or knocked out specific gene. Right? So here when they are removing that specific gene, it is used as an animal model in cancer research. Okay, so let us see how you are going to produce or generate a gene knockout mouse. So here the gene knockout mouse, you can see first you are selecting the mouse, you are selecting one of the mouse, then from that mouse you are going to take a blastocyst stage embryo, right? The blastocyst stage embryo is selected, and from that the embryonic stem cells are aspirated out. Then the aspirated embryonic stem cells are cultured in animal tissue culture laboratory and they are meant for genetic manipulation technique. Right? By means of genetic manipulation or genetic engineering, what you are doing, you are modifying or activating these embryonic stem cells in such a way that they disturb some specific genes. Right? So now they have got the ability to disturb the specific gene. Right? So then these embryonic stem cells are again introduced back into the developing blastocyst stage embryo. Right? So now these, see here you can see the genetically modified embryonic stem cells. Right? Now when they enter into the blastocyst stage, they start disrupting the gene. Okay? So now the disrupted gene is present in this developing embryo. Then it is introduced into the surrogate mouse. After gestation, it will give rise to the progeny, right? And now this progeny is said to be gene knockout mouse because that specific gene is not functional in that mouse. Is that clear? So here not functional means you are going to see all these are the genes, but I want this specific gene to be non-functional. So I cross it. So it is called as gene knockout. This particular gene is not functioning. Is that clear? So what is the use of this gene knockout mouse? Mainly in research, right? In order to study the cancers, heart attacks, arthritis, aging process, Parkinson syndrome and all. Right? So here let us see. We have seen the generation of knockout mouse. Then based on the research, again you have three types of knockout mouse. The first knockout mouse. See, knockout mouse means I am going to represent the complete set of genes. Only one specific gene is inactivated or knocked out. Right? So here the three types. The first type is constitutive knockout mouse. Second type is tissue specific knockout mouse. Third one is inducible knockout mouse. So let us see the constitutive knockout mouse first. So here now you can see the gene knockout means either you are inactivating the gene or you are replacing the gene. Right? You can see here, it is inactivation of the gene. This is the complete set of genes. This is the specific gene which I doesn't require. I want to inactivate. Right? So after generation of gene knockout mouse, with this process I have obtained this constitutive knockout mouse, in which in this mouse body that particular gene is not functioning. Is that clear? Next, coming to tissue specific knockout mouse. In tissue specific knockout mouse, again here this is if I take a single mouse. In the single mouse I want this gene to get expressed only in specific tissue but not in other parts of the body. That means here you can see in this specific tissue the gene is in active condition, right? It is functioning. Whereas apart from this tissue, all other parts of the body, it is not functional. Right? Within the single mouse, I want this gene to be active in some parts and inactive in some parts. For example, if we take in case of increased milk production. Right? So increased milk production, I want some protein to be present in that milk. So what I do? I introduce that gene of interest and allow that gene to get expressed in mammary glands only, not in other parts of the body, because mammary glands are only responsible for producing milk. Right? I want protein to be present in the milk but not in the body, isn't it? So I manipulate that gene in such a way that it go and expresses only in the mammary gland tissue but not in other parts of the body. In the same way, here this gene is specific in one tissue but in other tissues, in the complete body, it is not functional. This is called as tissue specific knockout mouse. Next, coming to the third one, inducible knockout mouse. That means in this knockout mouse, the gene is in active condition, right? It is functional. The gene will be like this in functional condition. When there is no inducer used, the gene is functional. But when I add the inducer, either the chemical inducer like toxins or chemicals, or a physical inducer, the radiations, right? What is happening? In presence of inducer, the gene is becoming inactive and non-functional. Whereas when I don't use any inducer, the gene is functional. So like that, based on the research, I can make use of constitutive knockout mouse, tissue specific knockout mouse and inducible knockout mouse. If you have any doubts, put it in comments. Subscribe for further videos.